So protection from infection is a multi-step process which starts with preventing adhesion of virus and bacteria to the epithelium cells. This prevention is done by normal flora in the gut and also in the lung, local chemical factors, as for example the antimicrobial peptides, mechanical factors and phagocytes. Once the bacteria has entered through the epithelium and there has been a small local infection, there are wound healing mechanisms to close the wound, or again antibacterial peptides, antimicrobial peptides, and the phagocytes of the immune system. Also gamma delta T cells will act at this stage. In a local infection that spreads in underneath the epithelium, the phagocytes, which are in this case macrophages and dendritic cells, will signal with cytokines, and they will also attract NK cells. The immune cells will then move to the lymph system, where the parts of the bacteria which have invaded the body will be presented to the T cells. These are presented by the antigen-presenting cells that have captured the antigens. The T cells will in turn stimulate antibody production, and cytotoxic T cells will kill the virus-infected cells in the mucosa. Also, activated T helper cells will recruit and activate macrophages in the mucosa upon infection. So, the immune system on the epithelium cells is consisting of the innate immunity and the adaptive immunity. And the adaptive immunity consists of the cellular immunity, which is T cell, and the humoral immune system, which is the B cell and the antibodies. The blue band of circles to the left represents the epithelium. The red little rods with cilia are bacteria. The first thing that can happen to bacteria is that we have antibodies to recognize it, and the antibodies is represented by that little green tree, and the antibody is triggering complement. And the reason why this little green tree is above the innate immunity and up in the top corner where the humoral immunity is because complement is innate immunity, but it is dependent to some degree on antibodies, which is the humoral immunity. The next thing you see popping up is the granulocytes. The granulocytes are part of the innate immune system, and they invade and kill unspecifically the invading bacteria. Next, we have the macrophages or monocytes, and they will also, of course, then eat. After that, what pops up is the NK cell in the far left corner, because this is innate immunity, but it's also cellular immunity. That's why it's so far down. The dendritic cells will also take up the bacteria. And then the dendritic cell moves to the adaptive immunity, where the dendritic cell has now the bacteria, the little red dots, presented on its surface. It presents to the cellular immunity, which is the CD4 and CD8 T lymphocytes. Also, the B lymphocytes are activated by dendritic cells and the CD4. So the CD4 is here in the picture, in between the DC and the B lymphocytes. The B lymphocyte will create the antibodies, and once there are antibodies around, the macrophages in the monocyte can then more easily take up the bacteria. And this is the last picture in the middle. We will talk more about the infection and immunity in the next lecture, which comes tomorrow. So, first I would like to make an introduction of the cells that we are going to talk a lot more about later on. So the first cell here is the enterocyte, and in the gut it is the intestinal absorptive cell. It has a simple columnar shape, and it lines the inner surface of the small and the large intestine. Number 2 is the goblet cell. It is also a simple columnar shape, and it has gel-forming mucins in its vesicles. The goblet cell is highly polarized. That means that the organelles and the nuclei is found on the the basolateral side. And on the apical side of the goblet cells, there are the mucin secretory granules. Number 3 is the Paneth cell. The Paneth cell is found at the bottom of the crypt of the villi, and we will come back to the physiology of the gut later on. The Paneth cell has large eosinophilic granules that occupy most of their cytoplasm. These granules contain several different antimicrobial compounds. Number 4 is the microfold cells, or the M cells, and they initiate the mucosal immunity by allowing for transport of microbes and particles across the epithelial cell layer from the gut lumen to the lamina propria, where the interaction with the immune cells can take place. Number 5 is the intraepithelial lymphocyte, the IEL, and these lymphocytes are found in the epithelial layer of mammalian mucosal linings, such as the gastrointestinal tract and the reproductive tract. However, unlike other T cells, the IELs do not need priming, and upon encountering antigens, they immediately release cytokines and cause killing of infected target cells. In the GI tract, they are components of the gut-associated lymphoid tissue, the GALT, which we will also talk about. Number 6 is the dendritic cell, the DC. So first we will mention the goblet cells. They produce the mucus, which is mostly water, and the mucus proteins, the mucins, we will talk about in the next slide. The mucus in the goblet cell granules expands in volume upon stimulated secretion. That means that these molecules are packed fairly tight inside of the granules, but as soon as the granules release, the mucus will expand very rapidly. It's like a pressurized can of shaming foam. On the little picture on the lower right, you see the goblet cells being stained inside of the small intestine of a mouse, and they are evenly distributed in the villis. So the Paneth cells, as described before, are found at the very bottom of the crypt enterocytes, below the stem cells. And these Paneth cells share some features with other immune cells, so mast cells and neutrophils. They can respond to bacterial cell wall components, and they will then secrete alpha defensins, lysozyme, and phospholipase A2, or PLA2, which cuts off a leg from the membrane phospholipids in the bacterial membranes. So now I'm quickly going to give you a little taste of what we're going to do tomorrow, which is talk about the immune system on the epithelial surfaces. Killing of microbes and infected cells by intraepithelial lymphocytes is a quite new field in immunology, and we will go through it thoroughly tomorrow. So just remember what I say in the following 2 slides. The intraepithelial lymphocytes belong to the class of cells called innate lymphoid cells. And from these cells, there are 3 clearly distinguishable subsets, and they defend against slightly different types of infections. The ILCs descend from the common lymphoid progenitor and turns into an ILC precursor. From the ILC precursor, the 3 subsets are distinguished by the 3 different transcription factors. So the ILC1, which is producing Th1 type of cytokines, is distinguished by the expression of Tbet, which is also a transcription factor expressed by Th1 T cells. The type of ILC expressing the Th2 cytokines is determined by this transcription factor GATA3. The ROR gamma T ILC is corresponding to the Th17 T helper cell. So upon activation of the ILC will produce their corresponding cytokine. However, while normal T cells are activated by priming and by the antigen-presenting cells. The ILCs are activated by cues or cytokines that come from the epithelial layer. IL-12 and IL-18 will activate the ILC1. The ILC2 is activated by IL-25, IL-33 and TSLP. And IL-1 and IL-23 will activate the IL-17. So what are the different ILCs doing when they sit there in the epithelium? The ILC1 is causing the macrophages to start to break down bacteria and virus, and it increases nitric oxygen production in macrophages. It confers immunity to intracellular bacteria and viruses. The ILC2 are implicated in allergy, but their actual function is to clear off helminths from the mucosal surface. The way to do this is that the ILC2 is causing a massive mucus production and also smooth muscle movement to get rid of parasitic worms. The ILC3 is activated by the epithelial cell layer and causes an increase in antimicrobial peptide production. And this is an effective way to reduce the number of extracellular bacteria. So just to do a very quick recap of the different cytokines. The interferon gamma is associated with Th1 function, and it is mostly a response to intracellular and viral infection. The Th2 is leading to a humoral immune response, and it's also in particular activated by parasites or helminths. The Th17 immune response is activated by extracellular bacteria, and it's also maintaining the mucosal barrier and contributing to pathogen clearance from mucosal surfaces. We will go back into this a lot more tomorrow, so you don't need to remember this right now. So another important part of the immune system on the epithelial barrier is the IgA. And IgA is produced by IgA-secreting plasma cells, which sit just under the epithelial membrane in the lamina propria. And once the IgA is produced, it will bind the receptor for IgE, the polymeric Ig receptor, on the basolateral surface of the epithelial cell. Then there is a vesicle created, and the antibodies are transported actively in vesicles from the basal side to the apical side. This process is called transcytosis. On the apical side, the receptor is cleaved off, and it leaves a component binding to the IgA. And this is a stabulatory component called a secretory component. This is not present on IgA found in blood. It will stabilize the IgA found on the mucosal surfaces and protect it from proteolytic cleavage. So the structure of IgA is quite interesting. It is, as opposed to IgG, a dimer, and it's joined by its R's end by the J chain. In orange here you see the secretory component, which is present on IgA, which is secreted on surfaces. We find IgA in all kind of secretions, and a great place to test for IgA concentration is in tears. The IgA is a poor activator of the complement system, and it also has anti-inflammatory effects rather than IgG, which may sometimes cause inflammation. So the way IgA is protecting against pathogens, it is that it can either act sterically, hindering a pathogen from attaching to the epithelial cells. This is often what is referred to neutralizing antibody. But it can also cause agglutination because it's a dimer, so it can bind to different bacteria with one molecules, and if you have very many IgA doing this, you have an aggregate of pathogens. This will stop them from reaching the cell layer.